I'm Anthony. These are my colleagues, Savannah and Ray. And through the Condor internship program, we looked at a study that dealt with parasite competition in barnacle populations. And this was conducted under the advisorship of our mentor, Caitlin Fong, and the principal investigator of the Marine Science Institute of UCSB, Armin Kuris.
Parasitism is the most common lifestyle of all animals, which makes it an important area to study. Barnacles are widely distributed and abundant organisms that have been intensively studied since Darwin's transatlantic voyages. Up to 14 species of barnacle have been infected with a parasite called Hemioniscus balani. Despite the fact that barnacles are such widely distributed and abundant, fewer than 20 studies have been made with its particular parasite. The barnacle we're studying is called Thalamus physis, and some recent research in Santa Barbara shows that some areas have up to a 90% infection. Such high infection implies that the parasite is important in the barnacle and rocky intertidal ecology. So we're interested in this project. We were interested in studying the abundance of multiple infections and the impact that it has on the parasite competition.
So this is our parasite. It's called Hemioniscus balani. This parasite is considered a castrator. So what happens, it usually swims under the barnacle and feeds on its ovarian fluid. Usually castrators are a 1-to-1 ratio, meaning every barnacle has one parasite, but in this case, multiple parasites could infect one barnacle, called a double infection. So when multiple parasites infect one barnacle, it brings up the question of competition and whether or not they will need to compete for limiting resources. And this brings us to our 4 main hypotheses. The first one is that the higher the infection is in an area, the more double infections there will be, as opposed to an area with less infections. Our second hypothesis is that parasites will prefer barnacles that are the same size as them, as opposed to smaller barnacles or even larger barnacles. Our third hypothesis is that when a barnacle has a double infection, it will produce an overall more eggs rather than a barnacle with only a single parasite living in it. And lastly, competition. So when a double infection occurs, they think that they will need to compete for limiting resources, making one parasite dominant over the other one.
Okay, and to test these hypotheses, we looked at a database of over 6,000 samples of barnacle that were collected from Southern California coast from Gaviota to San Diego. And we used a lab approach to determine whether or not the barnacles were infected and to quantify the amount of eggs within those parasites. So first off, we did the collection in the field at low tide, and those samples were brought back to the lab. And we have a little picture here just to kind of help you visualize how that went down. So we have some researchers collecting samples during the low tides with tools like hammers and picks and whatnot. And then from there, samples that weren't used right away were frozen for preservation. And finally, we used dissection microscopes and forceps to determine whether or not barnacles were infected and to quantify those eggs within the parasites.
So parasite detection, we have a simple method to determine whether this barnacle has a single infection or a double infection. All we do is invert a barnacle, and we take out all the tissue with some forceps, and as you can see the bottom picture, the red blob is actually the parasite. And once we're dissecting the barnacles, we have to consider the size of the infected barnacle, whether it's a multiple infection or a single infection, and how many eggs each parasite has.
Okay, so as mentioned, we were interested in looking at whether or not multiple infections increased as prevalence increased, prevalence of infection in the population increased. So here our x-axis shows the prevalence, and as it increased, the y-axis also increased as well with multiple infections. So we did a linear regression that shows a significant amount of data, and 85% of that is shown here.
So from data, we did find that the parasite was preferential to a certain size barnacle, and we used a model called logistic regression, which is capable of handling binomial data. And from that, we did find there was a size difference, as you can see on the graph. The median-sized barnacles were likely to have the most infections, and also were most likely to have multiple infections.
So we use the t-test to gather this data, which shows the number of eggs a single infection produces as opposed to a double infection. Even though you can see a difference in the heights of each bar, the difference is pretty negligible. So single and double infections produce roughly the same amount of eggs. And in the next graph, we show the proportion of eggs of the dominant and submissive parasite. The dominant parasite produces 78% of the eggs, roughly 3 times as much as the submissive parasite, which only creates 22% of the eggs.
Okay, so in conclusion, we saw that with higher prevalence came higher multiple infections, and we saw that the parasite preferred a median range size of the barnacle. And contrary to one of our hypotheses, we saw that the egg yield wasn't, with multiple infection, the egg yield wasn't any more than a single parasite. And even though that was the case, there was a dominant parasite that produced about 7% of those eggs. And so overall, these studies are important because they may be generalized to other parasite host systems. And recent studies show that there are infections that are increasing in the oceans, and studies like this can help to try and understand why things like that are happening. And yeah, so we'd like to thank our mentor, Galen, and the program directors, Wendy, Jens, and Dean, for all their help. Any questions?